All right, welcome back. Last we were together, we had put the DNA in the PCR machine and we're going to amplify our DNA. The next thing we need to do is to separate the smaller alleles, the mutation, from the bigger ones. To do that, we use a procedure called gel electrophoresis. Gel electrophoresis is a process of using an agarose gel, a sugar within a gel. It sets up kind of like gelatin, and it forces the DNA to go move between the agarose. The bigger pieces are slower to get through that gel, and therefore they don't run nearly as fast as the smaller pieces, and that causes or allows for separation.
Right, and between the two different alleles that we expect to see, the wild type CCR5 and the CCR5 delta 32, that's missing 32 base pairs. Even though 32 base pairs is not many base pairs, there's still a big enough difference in size that allows us to resolve that difference on an agarose gel. Because the mutated allele, the delta 32, smaller, it's going to travel through the gel farther than the wild type over the same amount of time.
Yep. All right, so what we need to do is make a gel like this.
Yes.
To do that, we need to assemble a gel casting box. So we have a plastic container, we have a couple of rubber stoppers, which are not always easy to get in. Got lucky that time.
You did. This process is very much like making Jell-O. So when you set out to make Jell-O, you pick the mold that you want it to set in.
Yeah.
Sometimes they have those cute little shapes. Sometimes you just use a square pan. This is the shape that we want our agarose gel to take.
And what we'll do is we'll put a comb in there when we have the liquid in there, and then that'll cool and harden.
Yes, and the comb will create, this comb has 10 wells that it will create where we can eventually load DNA into those wells. Great.
All right, how do we make an agarose gel?
We need agarose and we need buffer. The buffer that we use is called a 1X-TAE. It's a buffer that has enough ions in it to carry a current eventually.
Mm-hmm.
And we know that these gel molds hold a total volume of 30 milliliters. And we also know...
Milliliters?
Milliliters.
Oh, yes.
Yes.
The wells are all pulled on the order of microliters.
Yes.
30 mils. Listen to her.
We know that this entire gel mold holds a total volume of 30 milliliters. And we also know that we want our gel to be 2% agarose.
Real close to 2%.
Yes.
Mm-hmm.
So we're going to measure 30 milliliters of TAE and add how much?
You can stop the video right now and do a calculation. If 2% weight to volume gel and you have 30 milliliters, how many grams of agarose would you need? All right.
All right, the answer is 0.6 grams of agarose into 30 milliliters will give us close to a 2% gel. If we were doing this the volumetric way, we would first add agarose, the solid, and then bring the solution up to a total of 30 mils. But we're just hoping to get really close.
We will get really close.
Yes.
Within a half a mil.
Right.
All right, so we'll measure 30 mils of the liquid,
Mm-hmm.
and then we will measure 0.6 grams of the agarose powder.
All right, we want to make sure that we use what seems like an oversized flask because we're eventually going to boil this solution, just like you would making Jell-O. We want the agarose to get all the way into solution, and heat helps. So using a larger flask prevents the solution from boiling over.
And unlike gelatin, this will bubble when it boils and make a real big mess.
So big mess.
All right, it looks pretty awesome.
Not my first time.
Now, if you happen to get a mil or two extra, don't dump it back in there.
Mm-hmm.
Dump it down the drain.
Yes. Do not return liquid that has left the stock bottle back to the stock bottle.
All right, we have our volume.
To measure the agarose, we'll use an analytical balance like this. And the agarose comes as a solid in a container like this one. So I'm looking to measure 0.6 grams. First, use a weigh boat and hit the tare button to set the mass to 0 before adding my agarose. So at 0. And again, I want to avoid taking extra agarose out of this container, so I'm going to be really careful to try to hit right at 0.6. Right, with the 0.6 grams of agarose, you can add it right to our glass.
All right, next we need to get that into solution, so we're going to boil it. You can boil it in any microwave. I'm going to set it for 30 seconds, and that should be about right. All right, so I can see that it's still a little bit thick. It looks like it just boiled. I'm going to boil it one more time to make sure all that goes into solution. This time I'm only going to go for 10 seconds and start. All right, this is probably hot, so you might want to be careful carrying it. Maybe carry it with a paper towel or something. Now this will have to cool down. We'll let it cool until it gets to be about 55 degrees, and we measure 55 degrees roughly as the temperature where you can hold this comfortably. Now I have particularly dry hands usually, so I can take more heat, but this is definitely not 55 degrees.
So yeah, it's pretty hot.
Once it is cooled down, before it solidifies, we're going to add a special reagent.
This reagent is called CyberSafe Gel Stain. And this is what is going to let us visualize the DNA. So this stuff intercalates into DNA molecules, and it also fluoresces under UV light. So it will help us track how far our DNA fragments have traveled and then light up under UV light so that we can see them. And it's called CyberSafe because it is a safer alternative to other traditional chemicals that have been used to do this very thing.
Anything that intercalates into DNA could be a carcinogen. So be careful with it. We're going to need 3 microliters of that.
Right. I think it's ready.
All right.
You can see it has a color to it, so when I drop it in here, the whole solution will take on kind of an orangey color. And you want to make sure that you give it a chance to swirl it around to distribute the CyberSafe. And then we're ready to pour it into our gel mold. So when you're pouring a gel, you want to shoot for a depth of about 3 to 4 millimeters thick. And I think that we are there.
Looks great.
All right, so we'll let this solidify at room temperature. As it solidifies, it will get a lot more opaque looking like this one. And then it'll be ready to be used.
All right, when it solidifies, it's going to turn a white color. It does still have the CyberSafe in it, even though you might not see that red tinge anymore. So now what we need to do is remove the comb.
So you want to make sure that you pull the comb straight out. We don't want to rip any holes between the wells or below the wells. We want to keep those intact so that they hold our DNA well. So I always stabilize my forearms and make sure I have a good handle on the mold and the comb and pull straight up.
There you go, looks great. You can see it has a number of wells. It has a little ding there that didn't come from this. Might have come from somebody checking to see if it was solidifying too early. You can see from the side that those wells don't go all the way through. So we have some gel on the bottom. These will hold our DNA. Now we need to get these buffer dams off.
Right, so I like to score between the rubber stopper and the gel so that as you're peeling away, your gel doesn't rip. So I'll just drag my spatula between the two on the bottom. Nice.
There we go. Now we have a gel that we can put DNA in and run a current through it.
Yes. Yep, so we'll leave it in the plastic mold and carry it to the electrophoresis tank. So the notch on the gel mold fits right into the notches on the tank.
The wells are on the black side. This is the negative pole. Opposites attract, right? Or same charge repels. So we know that the DNA is a negative because it's got all those phosphates on its sugar phosphate backbone. So because it's negative, we can put it in a charged chamber and it will move away from that black negative pole toward the red pole. That's why we orient the wells on that side. Now, to get a charge going through here, it's not going to go through the air, so we got to figure out how to get it into our gel.
We're going to use the same buffer, that 1xTAE, to fill the electrophoresis tank, filling it just a couple millimeters above the height of our gel. I like to fill up both ends first. You can see it moving across. And it seems weird, but we actually want to add enough buffer to fill in these wells. When we pipette our samples of DNA, they're actually going to replace the liquid buffer that's already in the wells. So you'll be able to watch the buffer go over top and inside of all the wells.
Now, if you look from the side, you should see that the buffer is slightly above the gel height.
I still see a couple of spots where the gel's peeking through. All right.
We're ready to separate our DNA.
We're ready.
Now we need some DNA.
Where did we put it last time?
Well, DNA is most stable in the body, but if not that, then in the freezer.
All right.
So, last time when we were running PCR, we each did our own DNA, and we each did a control DNA. Students won't have to do the control DNA, but we do want to be able to look at different genotypes. The other thing we need is a DNA ladder. This is called a tri-di 100 base pair DNA ladder. And a DNA ladder really is a mixture of DNA fragments of known size. This particular one has fragments that are 100 base pairs apart. So it's got a 100 base pair fragment, a 200, and a 300, and a 400, and it kind of looks like the rungs on a ladder. So it's called a ladder. However, if we run our DNA in a lane right next to it, we can compare this measurement of the known amounts to our DNA, and we'll know what size our DNA is.
The ladder works much like a tape measure or a ruler.
Exactly. So, and CCR5, we know we had our primers 189 base pairs apart across that CCR5. Because the CCR5 delta 32 is missing 32 base pairs, it should have a band of 157 base pairs, 32 less than 189. Should be right in this region, but of course a different lane. Now it's convention to put your ladder in the first lane, and so that's what we'll do.
All right. We're gonna load 10 microliters of each sample and of the ladder into each well.
All right, our tank is set up. We're ready to add our ladder and our samples. Should we just start putting them wherever we want?
No, we should not. We need to have a record of which sample went in which lane. So I think it's a good idea if we lay that out on paper first.
I agree.
All right, so in lane 1, let's put the ladder. In lane 2 will be wild type control. In lane 3 will be the heterozygote control. Lane 4, my DNA. And lane 5, your DNA.
Sounds good.
All right.
And students should probably just put it right into that lab.
Yes,
That'd
I have
be a
no
great
for
thing.
that.
All right. So what we need to do is put the DNA down into the well. We can sort of see the wells. We can see the wells there. But we know that there's only a little bit of agarose below it, so I can't poke all the way through the well or it'll poke a hole in the bottom. I can trust that the liquid will drop down because each of these has not only dye in it, but it has glycerol in it, so it makes it more dense than the TAE solution. So stabilize your elbow, hold your wrist if you need to. It doesn't hurt to do that. Put it right down above that well, and then slowly put it in. As soon as you see that all of your liquid is out, stop depressing. Don't go beyond that first stop, because then you'll create an air bubble, which could get under your dye, and then reverse what the glycerol was doing by spreading it out, maybe dumping it into the next lane, and then you lose your data. Always change your tips. Now, the next person would load theirs. In this case, I will load the wild type. You see, we had 20 microliter reactions, and we're loading 10. So that if we do mess up, we still have one more shot to make sure that we get it right. So wild type goes in lane 2.
Alright, lane 3 is the heterozygous control.
All right, now we can clean up our work area here. Give ourselves some space. And now we have to start the machine.
Right, we want to apply current relatively quickly. The samples are okay in the wells like this because the glycerol keeps them sitting down nicely into the wells, but over time they will diffuse out. So we want to apply the current, get the DNA inside of the gel fairly quickly. So I'm going to make sure that my lid is attached to the power supply and the black lines up with black, red lines up with red, and snap it down without jostling things too much again because we don't want to mix our samples up. All right, that feels good. Turn the power supply on with the switch back here. And this is where we can control the voltage. So we are going to set ours to 95 volts. And hit the run button. How are we going to know if this is running, this is working?
That's a good question. We could hope for it, or we could look for bubbles coming off of the wires.
Yes. Looks very good.
All right, we'll let this run 45 minutes.
Right, and at this point, shortly after we start, we should see some separation of the dyes that were in our samples. They're also going to move towards the positive end.
That's right. So the negative charge is driving them away. So unlike DNA, you don't want to have negativity drive you. You would rather be attracted to the positive.
Great way to remember it. All right, our gel has been running for about 10 minutes, and if you look closely, you can see that the dyes in the green mix, the blue and the yellow, have separated. They're moving towards the red positive pole, and the tri-dye in our ladder in lane 1 here, you can see the 3 colors coming out of there. There's an orange and a light blue and a darker blue. So things are working well. Remember that these dyes are not DNA. These are simply additional dyes that are in the different mixes. Our DNA is only going to be visible under UV light, but these help us track the progression of electrophoresis.
So we see really nice separation. A couple of the dyes are already off of the gel, so we know that we've got nice separation. So we've been running at 95. We'll pause this. We'll stop it. Bubbles should stop. And we can even shut this off to be very safe. All right, now we need to pull it up without shaking it. So these can be held down while we pull up. That loosens that. Here's our gel. The gel might want to move on you, so make sure you are holding the edges. UV doesn't shine through plastic, so that's why this plastic protector is here to keep us safe. This machine will emit UV light, but if we lift it up, it'll shut the light off to keep us safe. It also means we can't just put our plastic thing on there, we have to slide the gel off. Get rid of the bubbles. I'll go shut the lights off quick. All right, so we're ready to take a look at this.
Right. Let's see. Moment of truth.
What were the lanes again? First lane was ladder.
Ladder.
Second lane was
Wild type control.
wild type. And then third lane.
Heterozygous control.
Fourth lane.
Mine.
Fifth lane.
Yours. All right. So first thing I notice is that the ladder looks really good. And this is a great indicator that we poured a good gel, that the fragments separated well.
Nice tight bands, a little bit wavy on the edge, but that's not a big deal.
Yep.
Nice tight bands in lane 2, 3, 4, and 5, which are our PCRs.
Yes.
There is a difference.
There is. It's very interesting.
So the first one is wild type,
Mm-hmm.
and we only see one band. Does
Yes.
that mean that they're haploid?
No, they're still diploid, still human, but both alleles must be the same. So we only see one band.
Oh, and they're just over top of each other. And
Yes.
that band is a little darker, so that makes sense. The
Right.
second one was heterozygous, so the third lane, the second sample.
Yep.
Heterozygous, we see 2 bands there. Do you see 2 bands?
I do see 2 bands there.
Nice.
Mm-hmm.
The bigger one, the one on the top, is the took longer to get through the gel, so that one's bigger.
Yep.
That's the wild type allele, and the bottom one is a mutation, so it's smaller. It went farther. It
Yep.
must be missing 32 base pairs.
It is.
The third sample, fourth lane, is
My DNA. It looks like
look at you.
I am homozygous
You
for
are wild.
the wild type. Huh. Very interesting. Okay. What about you? What do you see in your lane? The
Oh, I
last
think mine looks like lane 3, so it looks like I might be heterozygous.
Yes.
1 wild type and 1 delta 32 mutation.
Wow, very cool. So the other thing that we could recall is the DNA ladder and the fragment size is represented by the lowest two bands there. So the lowest band is 100 base pairs.
Yep, and the next one 200. Our primers were less than 100, so sometimes the primers can show up. We want to make sure these aren't primers.
Right.
So are these bands bigger or smaller than 100 base pairs?
They're definitely bigger. They're hanging right around that 200 base pair fragment from the ladder.
Which is perfect because we thought the wild type was going to be 189.
Right, the bands are showing up right where we expected them to. All good signs.
Very good. The other one's kind of in the middle of the 100 and 200, and we expected that at 159. So that, or somewhere in there, real close
Right.
to that. So 157 was it?
Yep.
And that looks right too.
Yes, very cool.
Exciting. It does also show that we are human. If we saw no DNA, would that mean we were a zombie?
I mean, I would be tempted to think so. But no, the other option that we could have seen in any given lane is a failed reaction where we don't see any bands.
Right. So the DNA could have poked through a hole through there, or something went wrong with our PCR. We forgot to add one piece, one of the important things in it.
Right, or even something with the DNA extraction. So there are several points in the project where something could have gone wrong, but we had some built-in controls, so we're able to figure out what's going on here.
All right, looks great. All right, let's grab a quick picture of it.
All right, I'll snap a picture here. So we want to take a picture before we expose the gel to the UV light for too long, because our bands will fade over time if they are under too much UV exposure. So now we have a picture. We can use this to analyze the data further to determine how many alleles of each type we have for our own samples and for our classmate samples.
Excellent. So we'll be able to calculate allele frequency in this population.